Within the project LiveScience, the University of Applied Sciences of Western Switzerland has to integrate living cells as biosensors, which are developed, chosen and tested by other groups. In a first step, we have chosen the biosensors which are suitable to be implemented in a small, portable and standalone systems. The first choice was genetically modified bacteria engineered by the group of Jan van der Meer of the University of Lausanne. In contact with arsenite in water, these bacteria produce a GFP, which stands for green fluorescent protein. Water collected by the standalone system is brought to a microfluidic chamber in which bacteria are concentrated. The amount of GFP is a function of the time and the concentration of arsenite. Previous studies and reference comparison on boards allow the measurement of the arsenite concentration in water. This is done by exciting the GFP with the blue light and measuring the green fluorescent light after blocking the blue excitation light with an optical filter. Repeating these measurements every 5 minutes allows the calculation of the concentration of arsenite. The results are sent via a GSM module to a mobile phone. Other groups are working on different biosensors such as electrical conductivity on epithelial tissue of C2b-BE1 cells developed by CSEM.
Within the LiveSense project, we have developed a device that allows us to screen for cell toxicity without using any microscopy or biochemical assays. Instead, we are screening the impedance of the cells with electrodes. And we have actually found out that by screening different frequencies, we can obtain different information about the cell status. A cell that is exposed to a drug first undergoes morphological changes that can be monitored with the low frequency impedance, and the later occurring cell death is observed at the high frequency impedance. For this, we seated our cells in a microfluidic chip in a cage structure that allows a very efficient medium renewal and guarantees small diffusion distances of the drug that is thereupon perfused. After drug exposure, we observed immediate morphology changes while the cell viability remained high and only decreased after some lag time. We estimate our technique ahead of conventional viability assays because we don't need the cells to die to sense toxic effects.
With our label-free screening platform, we see a clear difference in drug response between doxorubicin-resistant breast cancer cells and their parental cells. When 20 micromolar doxorubicin was applied, the drug was strong enough to kill the wild-type cells within 1 day, while there was no effect of the drug on drug-resistant cells after 2 days. After applying different drug concentrations, the dose-response curves show around 500 times higher IC50 value for drug-resistant cells compared to the wild-type cells. These findings verify that with our cell-based platform, we can monitor the drug response of breast cancer cells at earlier and more progressed stages of the disease. Our non-endpoint assay could find a promising application in the field of personalized.
In the LiveSense project, we studied the cell's reaction to toxic samples. One of the powerful techniques for evaluation of the cell's reaction is to monitor the metabolism rate of the cell. The main metabolites in a cell culture are glucose and lactate. We know that cells usually consume glucose and produce lactate. So if we can measure these two metabolites simultaneously, after exposing a toxin, we would be able to see the changes in the metabolism rate. Here is a photo of the microfluidic cartridge we have developed in our lab for inline measurement of glucose and lactate inside the cell culture medium. The cartridge is fabricated by rapid prototyping on a glass substrate. As we apply a small volume of a cell culture medium into the cartridge, the sample flows through the enzyme microreactors and then is directed over the downstream microelectrodes. The microreactors are SU8 pillars on which glucose oxidase and lactate oxidase are immobilized. The pillars are structured to increase the surface area for immobilization of the enzymes. In a cell culture medium, the concentration of glucose and lactate were measured over a period of 5 days using our microfluidic cartridge. As it is shown in this graph, glucose consumption and lactate production of the cells could be precisely detected over time. We also monitored the metabolism state of the cells 24 hours after exposing them to a toxin. As we can see in this graph, we could detect a decrease in the production rate of lactate and the consumption rate of glucose due to the effect of the toxin. Another important aspect in the LiveSense project is the sample preparation. We need to adjust some physical and chemical parameters of the samples before they come into contact with the cells. Here is a fully automated portable system that we have developed for adjusting pH and osmolality of the river samples.
The idea behind our project is rather simple. Any living cell sitting on a surface applies forces, and as soon as pollutants interfere with the vitality of the cell or finally even kill the cell, the forces that a cell can apply to the substrate are reduced. So a broad range of pollutants can cause these type of effects. Pollutants, for example, that impact the membrane functions, pollutants that impact the contractility of cells, motor proteins, the cytoskeleton, and certainly also other factors that interfere with the fourth generation of cells can be monitored.
To fabricate biosensing nano-pillar arrays, we use molding process. We first fill SU8 photoresist into PDMS mold, then after degassing, UV curing, and unmolding. This electric nano-pillar array was then made. We then seeding cell on top of it. Cell can apply mechanical force to the pillars and deflect them. Here we will show how the cell move around on top of pillar, then cell deflect pillar one after the others, and altogether are 2,000 pillars per cell.
If the cell gets exposed to a toxin, you see how a living vital cell is turning into a lame duck within about 10 to 15 minutes. So with this we demonstrate that we can indeed use these nanopillar arrays to measure toxins in the environment of cells. So now after we've demonstrated that we can engineer very robust platforms for future technological applications, the next few stages will be for us to integrate such a system into long-term monitoring devices and to also work on simple readout schemes for these post-deflections. Thank you very much.
In our lab, we are developing bacterial bioreporters to detect environmental pollutants. For example, in Bangladesh, a bioreporter system has been successfully applied in the field. These genetically modified bacteria react to the presence of a target compound like arsenic and produce an easily measurable signal. We can rapidly and easily measure the bioavailable fraction of pollutants at a low cost.
So for LiveSense project, we are developing a microchemist on chip, and the goal is to actively maintain bioreporter cells in a microchamber for 1 week. Then, using microfluidic valves, every time we need to perform a measurement, we expose part of these cells to arsenic. This is the LabVIEW program that controls the opening and the closing of the valves. And now we connect all the tubings. We inject the cells in the chip we are filling the chamber with the cells. Here you can see the cells that are coming. When we want to give some nutrients to the cell, we open the valves before and after the chamber. In the measurement zone, there is a filter that retains the cells, while the arsenic solution can pass through the filter holes. The fluorescence intensity produced by the cells increases with time, and after 180 minutes we can determine the arsenic concentration present in the aqueous solution.
Yes, yeah, I'm going a little bit closer. Okay.
Like this?
Yeah, it's fine. Yeah.
In the LiveSense project, the Laboratory of Physical and Analytical Electrochemistry has been involved in the preparation of multisensory platforms, for instance, in the development of oxygen and conductivity sensors. More recently, in collaboration with the team from the University of Lausanne, we have developed an electrochemical methodology for the readout of arsenic-sensitive bacterial bioreporters. The principle of the arsenic bioreporter is based on a genetically engineered bacterial cell that in presence of arsenic-3 is able to generate a signal that can be read electrochemically. In this case, this signal is paraminophenol that can be electrochemically oxidized, and the current generated from this process can be correlated with the concentration of arsenic that is sensed by the bacteria. The formats that we are using to perform this assay is by using this very cheap and portable microchip that contains 16 independent electrochemical cells that allow us for a high throughput analysis. What we need to do then is just to add the sample, the substrate, and the cell suspension in each well, and basically just locate it in this portable potentiostat and to start to record the signal that is obtained from each cell. External calibration of our methodology has been performed in tap water, and we have shown that the limit of detection is equal to 0.8 ppb, which is one of the lowest limits of detection for arsenic by using bacterial bioreporter. Additionally, we have validated our methodology by quantifying the concentration of arsenic in Swiss groundwater that has been previously quantified by atomic absorption spectroscopy at the Cantonal Laboratory of Sion. We have seen a very good agreement between the concentration of arsenic determined by the bacterial bioreporter and the atomic absorption spectroscopy methodology, showing that this is a real alternative for quantification of arsenic in real samples.
At CSEM, we aim to use transepithelial electrical resistance measurements, or TEER measurements for short, to monitor the integrity of a monolayer of cells. In comparison to traditional cytotoxicity, TER measurements are seen as a non-invasive and non-specific way of monitoring the integrity of a monolayer of cells or the toxicity of a sample. Traditional methods for taking TEER values were done using green STX2 chopstick electrodes. A resistance value was seen on an EVOM multimeter. This method only allows single measurements to be taken. We are aiming to move away from static measurements on polymer membranes towards taking continuous measurements on ultra-thin porous silicon nitride membranes in an integrated fluidic system. Here you can see the PDMS bioreactor, polycarbonate membrane holder, two black clamps to hold the system together, and the gold electrodes for taking the TEER measurements. Cells are cultured in 6-well plates using a polycarbonate holder for the membrane before being transferred into a PDMS bioreactor. Good cell growth and differentiation is seen over 21 days in order to obtain a tight monolayer of cells with high resistance values.
The PDMS bioreactor has a distinct apical and basolateral compartment. This is a requirement for TEER values. The fluidic system is powered by a peristaltic pump at a flow rate of 200 microliters per minute.
TEER measurements are taken at 10-minute intervals for 10 seconds at a time. A drop in TEER is seen as indicative of the early stages of a cell's response to a toxin. This measurement system provides an alternative in the early detection of non-specific toxicants.